Whenever we deal with organisms, one of the things we have to deal with is we have to deal with the concept of taxonomy. And taxonomy comes down to how do we group these things? How do we put them together in a way that we can actually compare and see what's more related to what? And, how do we do all of this? So let's take a look at some of the terms we use and the way this works. Some of the terms. Morphology. Morphology comes from the Greek word morphos, which means form. So we're looking at the study of form. What does the organism look like? And based on what it looks like, how does the form come together? And what are we dealing with as far as form goes? If we look at humans, humans have 2 arms, 2 legs, a head, and a trunk. Some organisms also have 2 arms. Humans stand on their hind legs. Other organisms may have hind legs, but they may also use their arms as far as support goes. Some organisms have 4 legs. You take your pet, and chances are your pet's going to have 4 legs, which means it's got a little bit different form. So when we look at this, we deal with form. What does it look like? We've got 3 different mushrooms here, and you can see that the 3 different mushrooms have a tremendous amount of variation between them as far as form goes. And based on this, we can use these forms to break them apart. We have taxonomy. It says taxonomy is the naming and classifying of species. In taxonomy, we figure out what a species is, we give it a name, and there are certain guidelines that we have to use in order to be able to deal with that. And therefore, it becomes very important. We have classification. Classification says we're going to take those things that we've named and we're going to place them into predetermined groups. Sometimes we find out the groups aren't too good and we reassign them. So in classification, it becomes very, very important to understand where we're going. We have what we call phylogenetics, and phylogenetics is the new big thing. And basically what you're looking at is you're looking at evolutionary relationships through genetic relatedness. Which came first? And how related is something? If you look at it, all humans on planet Earth came from a single female that lived between 100 and 150,000 years ago. But if you look at the differences we've got between us, it is absolutely amazing. So when we look at morphology, everybody's kind of similar, but when we look at, phylogenetics, certain groups are very distinctive from other groups. So it becomes very important to look at that as well. And then we have the definition of a species. The definition that I like is a group of organisms capable of interbreeding and producing fertile offspring. So it says you've got to have things that have the same form. They've got to be able to interbreed and produce fertile offspring. This goes back to the concept that you all know what a horse looks like. Male horse, female horse to get together, they have little baby horses. You all know what a donkey looks like. A male donkey and a female donkey get together, they have little baby donkeys. If you take a male donkey and a female horse, you get a mule. The mule has characteristics of both, therefore it's a hybrid, but the mule itself is sterile, so it cannot produce. And this concept of hybrid is something that we often deal with, but they are not what we would call true species. So therefore, we look at it as just a hybrid, not a species. So a species has to be able to interbreed and produce fertile offspring. And then we talk about cladograms, and we'll use cladograms to show the way things are different, especially when we get into the taxonomic part. And it says that cladogram is nothing more than a tree-like diagram that shows evolutionary relationships. Which came first, and what's more closely related to what? And these are very, very helpful when we look at them. When we deal with organisms, we look at a lot of different ways of putting things together. We know what a species is based on the concept of the species. Similar species are put together in a genus. Similar genera are put together in families. Similar families are put together in orders. Similar orders are put together in classes. Similar classes are put together in phylums. Certain phylums are put together in kingdoms, and similar kingdoms are put together in domains. So when you're looking at something like a domain can be very simple in the similarities. This particular one says bacteria. Bacteria means that it has to be a prokaryotic cell. And that's all. Right? So we have no other specifics in there. If we look at some of the other parts in here as we get down, we find more and more similarities in there. Now, a couple of things to note. The species name is composed of two words. It's composed of the genus and what we call the specific epithet. And we underline this, or we italicize them. So you can see it's written in italics. The genus is also written in italics. We often put the authors down afterwards because it lets us know who we've got or where it came from, and when was the last time it was changed. So we're gonna take a look at the way this concept works as it relates back to different organisms. Now, this organism that we're going to look at in here, you can see we got a lot of different things in here. And we're going to start out looking at a plant-based organism. And it says the first thing we have to understand is what domain are we dealing with. It says we're going to be dealing with the domain Eukaryae. So Eukaryae means everything is eukaryotic. So the first thing we have to do is we have to remove from this picture, which goes all the way from a bacterium down to a donkey, we're going to take anything that's not eukaryotic out. And all we lost were the bacteria up in the upper left-hand corner. The next thing we're going to look at is we're going to look at the kingdom, and in this particular case it says Plantae. So we're going to take away anything that is not considered to be in the kingdom Plantae, which means what did we just lose out of there? We lost the fungi, we lost the algae, we lost the animals. You see that? And now we're just dealing with plant things. And as we get in through botany, we'll look through plant things. If we were looking at microbiology, we would look at things in the prokaryotes. If we were looking at certain areas of biology, we might look more towards the animals. Next concept, we're looking at a phylum. A phylum is a subdivision of kingdoms. And in this particular case, it says we're looking at the angiosperms. Angiosperms are flowering plants. So let's see what disappears with the angiosperms. Oh, that whole group, what do we have? We had pine trees, we had moss, other things like that. Those are not angiosperms because they are not flowering plants, but all the rest of these are. Then we go, within that, we're going to look at the monocots. So we're going to take everything that is not a monocot out of this, and we're going to see what we get left with. All right, now I'm looking at this and I'm going, the upper left-hand part is Magnolia. We've got some dicots in here. Some of them have pretty flowers. Some of them are all varied all over the place. So, we just lost a whole bunch. We lost the Magnolias and we lost all the dicots. And what we're left over with now is monocots. And what do we see in here? We see things like a daffodil, bananas, an orchid. That's an iris. Cattails, corn, grasses, and we've got a lily down here on the bottom. The next thing we're looking at the order Asparagales. Now you can see the basal root of asparagus in here because it's an Asparagales, and we're looking at things that are going to have those specific characteristics. Now let's see what we lose. We lost the grasses, we lost the bananas, but what we're left with is the gladiolus, the daffodil, the orchids, and the iris, because they're all in the Asparagales. It says next, we're looking at the family Amaryllidaceae. So if it's not in the family Amaryllidaceae, it's not going to stay, which means our orchid is about to go bye-bye. Oh, we also lost the other flower in there. Now we've only got 2 left out of everything we started out with. It says next we're looking at the genus Narcissus. Oh, and all we've got is the daffodil. And the daffodil is in the species Narcissus pseudonarcissus. So that's how we take these and we break them apart and we build up in a reverse manner. So when we look at it, that's what we're dealing with as far as taxonomy goes. Now, if we're dealing in microbiology with things like bacteria and stuff like that, sometimes we don't have the ability to look at a lot of detail. So what we often do is we often do biochemical testing. And sometimes this becomes important in higher organisms as well. We're looking at what are similarities. These are sugar fermentation tests. That's a urea test. Down here we've got, oh, what is this? This is Sims media. Sims media is going to look at motility. It's going to look at indole production. It's going to look at sulfur reduction. So we do have variations there. We're also going to talk about cladograms. What are we doing here? We're looking at different units within the plant kingdom. And it says we're going to look at bryophytes and pteridophytes. And you can see here what we're doing is we're building it up and we're looking at differences. It says the bryophytes do not have a vascular system. Put in a vascular system, we're looking at the pteridophyta. We put in seeds, we get up to the gymnospermy. We put in flowers, we get the angiosperms. So when we deal with these, clads become very important as well because we start to see a little bit of difference. And we will look at taxonomy and the way we break things down throughout the rest of the course. So therefore, taxonomy becomes very important because it gives us a way to be able to group together similar things so that we can study things and we can relate to them in an easier manner.